maybe I can start. One thing that I am very impressed about your presentation and also your presentation is this pathophysiology of inflammation and all these disorders. Do you have any idea about if the innate immune system is playing some role in this?
Yeah, so we do think this is the innate immune system that's being impacted. Yes?
Yeah.
Short answer.
Yeah.
So these are, so I'm not a neuroimmunologist, I'm a psychiatrist and a cognitive neuroscientist, but we believe that these effects are mediated by the toll-like receptors in the innate immune system.
Yeah.
Yes.
Because inflammation is everywhere, in obesity, in chronic and non-communicable disease like diabetes, and even in my area, hypertension. Inflammation is coming to explain several things. So we are in the era of inflammation. What is your experience about it?
No, I think it's very much true, and what we thought in the past that didn't modulate the brain, now we know that modulates the brain. So inflammation is one of the clues into to understand that. We know that inflammation changes cognition as well. And so diseases such as obesity is influencing the brain through inflammation. So there are many connections that are actually outside the brain that is more linked to the general health. We actually, together with Nudel, we also work with ACE, that is much more from your area. And interesting enough, there are several connections between ACE, angiotensin converter enzyme, that is from the cardiology, and even genotyping it, and schizophrenia. If I knew you were here, I was going to present something related to that. So there are plenty of pathways, I believe.
And it may be the case that inflammation is not maybe the best word to characterize the role of these molecules in the brain. Clearly, there is this interaction between the peripheral immune system and the central nervous system, so that if there's inflammation in the periphery, it can have an impact on the function of these little sticky molecules in the brain. But it seems that the molecules themselves are really serving, at least in part, a separate function. Their role is to help guide the organization of cortical circuits during early brain development, and to manage the number and the strength of connections between neurons in adulthood. So it's a system that the brain borrowed from the body in order to help to organize itself. And I think perhaps by coincidence or perhaps by design, there's this interaction that we didn't know about between what's going on in the periphery and what happens during brain development.
And the last one, very simple question. What do you guess about the percentage of role of genetic environmental in schizophrenia? What is
the role?
The role of each one. What is the relative role?
Well, so the concordance rate of identical twins is 50%. So there's a heritability of 80%, so it's very genetic, but there's a concordance rate for identical twins of only 50%. So the environmental risk factors are really key. And in the way that we're beginning to learn that there are multiple genes of small effect, and presumably a lot of sort of epistatic interactions that have to happen between a range of risk genes in order to increase your genetic risk, there's clearly a really important role for the environment as well. And these so-called neuroimmune mechanisms might represent a common pathway through which the environmental risk factors impact brain function and development and interact with the genetic risk factors. So.
I absolutely agree. Just to give you an example, most of the genes, the polymorphisms of the genes associated with schizophrenia has a relative risk of about 1.2. When it's very high, it's about 2. When you talk about urbanicity and migrant immigration, they get up to 4. And the idea is when you combine some of the polymorphisms that predispose you to schizophrenia, for example, and you link it, for example, with smoking cannabis, you have a synergistic increase of relative risk. So that's the kind of interactions that we expect to see in the future. But we are here talking about a huge amount of data. It's not only one gene and one risk factor. It's
I believe there are 3 questions from the floor. One, please. Identify, make the question.
Good morning. My name is Raquel. I'm from Hospital Syro-Libanese, and I wonder to know more about the free water studies that Dr. Carter told. And for Dr. Bressan, I would like to know more, like how he interprets the correlation between the decrease in activation in the refractory patients.
sorry, decrease in activation of?
Yeah, that you show, the decrease in activation in the refractory patient in the brain.
I think it was cortical thickness, maybe.
Oh yeah, it was. No, it was cortical thickness, not activation.
Just
The
cortical.
images are beautiful. The amount of what we know, it's not so much. So sometimes we're talking about activation, and sometimes we're talking about cortical thickness. It's structural. So it's thickness of the cortical layer.
And how do you interpret this? Is because of that they got the schizophrenia, or this like the result? How do you interpret it?
It's the chicken and egg thing. That's why we're moving towards the early stages before the disorder itself. When you get a resistant patient with changes in dorsolateral prefrontal cortex, for example, you never know if it's chronicity that is predisposing to treatment refractory mass, or it's let's say a brain change that would predispose in the future. So I can't answer. So what we hope is when we put the sample together, we can, although we are not following up the patients because it's a long way, you can compare, let's say, the dorsolateral prefrontal cortex in different stages and see if the refractory patients do have thinner layers than, let's say, in the first episode.
And your question about free water, so we use a multi-shell diffusion acquisition. The shell refers to diffusion gradients that can be tuned so that they're differentially sensitive to the anisotropic component of water in the brain, which is generally in axons and cells, and the isotropic component, which is in the tissues. So typically diffusion MRI has one B value and is optimally sensitive to the anisotropic water. But if you use multiple B values, you can separate out those two components. So this was a custom-developed pulse sequence that we are using at UC Davis.
And it's able to do in humans and other animals?
Well, so it's able to do in humans, and we will be doing this in our macaque model, and we will probably explore being able to do it in our mouse model as well.
Thank you.
Who else?
Morning, Raul Maranhão from the Heart Institute. I'd like to know if you have both or two, if you have any relationship data on lipid metabolism and schizophrenia as you have in Alzheimer's disease of APOE4.
Well, you know, I think our medications cause metabolic syndrome through mechanisms that we don't fully understand. And this is a big challenge. People with schizophrenia have shortened life expectancy of about 15 years. And that 15-year shortened life expectancy is partly because suicide is common. About 10% of people with schizophrenia commit suicide. Smoking is common, and this increases cardiovascular risk. But then our own treatments also contribute to cardiovascular risk. Whether there's an innate form of sort of metabolic syndrome that's associated with the illness itself and not caused by medications, people have speculated about that, but I think there's no question that our medications contribute.
Thank you.
If I can complement, there was data already showing that before the antipsychotics there were changes, more diabetes and obesity. And you never know if it's a consequence of negative symptoms, that's so you don't exercise and then you kind of increase your weight. But I thought you were asking more related to predisposition, and there is quite a bit of work from Gattas, from USP here, that works with phospholipase A2, so that he measures in the periphery. And I'm not very aware of this research line, but for sure is one of the, it does make sense, one of the, let's say, lines of research.
Okay, thanks.
Thank
Question here.
Aqui, ó. Por favor, aqui na frente.
Did you have a question?
Hi, morning. My name is Cláudia Del Fava. I work in Biological Institute, São Paulo, Brazil. My question is, are you interested in studying in your projects of research human patients who have lost the right frontal cortex, I want to say malacia, as cause of cerebral hemorrhagia, and these patients are maintaining a very right and chronic monocytosis in their blood, and they are showing psychiatric disorders and problems in cognition as well?
I think the answer is yes. These patients sound very interesting. If I understood your question correctly, they sound very interesting. So these are right frontal lobe injury patients?
Yes.
Okay. Very interesting. So let's talk in the break.
Yep.
Here.
Benny Osborne. I'm at the University of California. I'm wondering if there's any role for understanding the development of cytokines or the ontogeny of cytokines, and when some of these may have alterations in the tracking of the developing neurons.
Yes, that's the benefit of the mouse model. So Kim McAllister has already done a very extensive developmental analysis of cytokines in general in both typically developing mice and in mice with maternal immune activation. It's so complex, it's incredibly complex. But at the cellular, she's also looked at the neurons from those mice in slice and looked at spines and used knockouts to sort of unpack the molecular pathways that mediate the effects of maternal immune activation. So there's a set of pro-inflammatory cytokines, particularly IL-6 and some others, that play a key role in regulating spine density and and synaptic strength in the slice. And the molecule that seems to mediate that is MHC. Yes, there's a lot of important basic science that can be done that can give us some real insights into how this all plays out at the cellular level. Yeah.
Oh, I'm happy.
Okay, so we have a very exciting session, and we thank again the speaker for the excellent presentation. And we do hope some cooperation can start from this presentation, some curiosity about complementary works in these groups. Thank you very much. Now we have coffee.
Thank you.
Coffee break.
Thank you.